The overall goal of this experiment is to determine chloride selectivity of new light-gated ion channels.
This method can answer key questions in the biophysics of channelrhodopsins, such as photocurrent amplitudes, kinetics, and ion selectivity.
The main advantage of this technique is a direct readout of biophysical channel activity that is fast and easy to replicate.
Generally, individuals new to this method will struggle because each step of the experimental procedure, from the preparation of Hec cells to the recording of photocurrents, has to be optimized. Seeding and transfection of the Hec cells will be demonstrated by Altina Klein.
To begin this procedure, weigh the solid components for 100 milliliters of the intracellular and 500 milliliters of the extracellular buffered solutions in separate beakers. And then add the respective amount of the prepared stock solutions. Next, add ultra-pure water and carefully adjust the pH using acids and bases. Then, adjust the osmolarity with glucose to 290 milliosmoles and 320 milliosmoles for the intracellular and extracellular solutions, respectively. In this procedure, place up to 3 lysine-coated glass cover slips side by side in a 35 mm Petri dish. Then, seed 1.5 times 10 to the 5th Hec cells in 2 milliliters of standard DMEM supplemented with 10% FBS and 1 micromolar retinol in each dish. To transfect